Okay, so I've had several students ask me about the sections or the chapters of the study guide to be turned in on exam day. Of course, here the idea is that you study, that the study guide helps you study. So you're required to always turn in up to the chapter that will be included in the text. So we're finishing today what Rock said, this chapter, and there is the last chapter in your study guide that you have to turn in is the What the Rock Said chapter. This one that is entitled this way. Okay, try to entitle my my presentations as the chapter so it helps you understand. Okay, so on exam day, you all whoever decided to do the homework will turn it in with me, and then we start the exam. Okay, and remember it has to be handwritten. You cannot turn in typed answers. I will not take it. Every time I have somebody say, oh, but I didn't understand that. Okay, so I try to repeat it. All right, so exam is Monday for you guys. So we have the whole weekend to study. Try to study these days, not the last day only, not the last minute study doesn't work, I guarantee you. Any questions about the exam or anything? So the format of the test is mostly multiple choice questions, mostly. But there are a couple of like short answer questions and maybe fill in the blank, things like that. All right, so last time we were talking already about fossils, right? Kind of we're already kind of halfway through. We talked about the type of fossil formation, right? We talked about mineralization, and we talked about mold or cast. And I believe we were right here, trace fossils, right?
You're talking about absolute dating.
Oh, so we were more advanced. Okay, good. All right, so we already talked about all the types of fossils. We talked about relative dating of fossils. Remember, this is a relative dating where I look at the position in rock layers where some fossils have been entrapped. And therefore I know that because there is a sequence, a succession from the bottom to the top, I know which ones are younger, which ones are older. The older ones will be on the bottom. So we talk about succession. If we're talking about animals, we call it faunal succession, and the layers are called strata, singular stratum. And the one thing that we gather from this knowledge of how the younger fossils are on top and the older on the bottom is that life goes in time becomes more complex. Organisms evolve more complexity. And that's a general trend. Okay, so yeah, now I remember I actually asked you guys, in order to kind of not spend too much time on this radiometric dating or absolute dating, I asked you to review the structure of atoms, okay? Because we're going to talk about that today in order to understand conceptually really how absolute dating works. So absolute dating is a quantitative method, so it gives you a number, it tells you approximately how old, exactly in terms of numbers of years, is that fossil. This technology is based on the knowledge of isotopes. An isotope is a form of an element that is not quite as stable as the most common or normal element that is present in it. It happens for many elements that they have some, let's call them cousins around. They are the same element, but yet they have a little bit of a difference. And the most important difference is that you can consider them like the crazy cousin. Who has a crazy cousin? They're a little bit crazy. They're unstable, we say. Okay? Now, if you have reviewed the structure of an atom, you may recall that an atom has subatomic particles to it. And so, as very simplistically, we can represent an atom with a core, okay, in which we find what? Protons and neutrons, these two types of subatomic particles, and the electrons are found kind of out in the space outside, okay? The very simplistic representation is the Bohr model where the electrons are orbiting around. This is not really true. That's what we mostly learn in school, right? But for the purpose of understanding radiometric dating, it's good enough. Now, what really defines an atom is this, is the number of protons, okay, which are positively charged in the nucleus. So if I ask you, there is a tabular representation of all the naturally and non-naturally occurring elements, periodic table. If I were to ask you, I give you a periodic table, the periodic table does give you this information on it. All periodic tables do. It tells you how many protons each element has. So I can ask you, go on the periodic table and find me the element that has one proton. There is only one. Okay, there's not more than one, because if another element also has only one proton, it's the same element. It's not a different element. So that's the something that gives identity. And in this case, if I asked you, there is an element that has only one proton, it's hydrogen. Okay? And actually hydrogen is the only element that has no neutrons in its nucleus. No neutrons at all, just one proton. Now, let's talk about hydrogen. It's kind of like easy. So the normal form of hydrogen in nature, the stable form, has a proton in the nucleus, nothing else, and an electron. Its number of protons and electrons are always the same. Now, there are a couple of cousins around, a couple of cousins of hydrogen. They're called the deuterium and tritium, okay? Which have respectively 1 and 2 neutrons. So you see that this element, okay, these two elements, and an electron, of course, that orbits around, let's say, so you see the element is still hydrogen because there's one proton, But this one has a neutron. The stable normal form of hydrogen has no neutrons whatsoever. This one even has two. Now neutrons are kind of heavy particles. So whenever that happens, whenever an element comes into a form where it has additional neutrons, we call it heavy. It's a heavy element, or it's an isotope, we can call it, there's another thing, because it has extra particles that have a weight to them. Okay, simple as that. And the other important thing, as I told you, is these are crazy. They are unstable. They have extra energy to spare, which they emit constantly. Okay? And by emitting those energy and particles as well, it's kind of like they try to get rid, in a way, of these extra particles. They try to reach stability, because the stable form is this, not that. In doing that, we call that process decay. So they decay in time, okay? And the rate of this decay depends, is very unique to each isotope. Not all isotopes decay at the same rate, with the same velocity. Some do much faster than others, okay? So that's the general concept, okay? So we're talking about isotopes. So elements, forms of elements that are unstable and have extra neutrons, okay? And pretty much in nature, many, as I told you, many elements have isotopes, including carbon, okay? This is what I just told you, okay? And a radioactive atom emits particles and energy to try to reach a more stable state, okay? And as much as many elements in nature have isotopes, carbon also has an isotope. The normal form of carbon, the one that we see in the periodic table, is known as carbon-12 because it has actually 6 protons and 6 neutrons added together gives you a number 12. So carbon-14, it's a different form, it's an isotope that has 2 additional neutrons. So it's a heavy element. Now, in general, when we talk about decay and radioactive or radioactive isotopes, unstable isotopes, we call the starting element isotope parental, and whatever it ends up to be after decay, daughter or stable. Okay, so this is just general introduction, okay? I told you. Now, carbon-14 is the isotope, the unstable radioactive isotope form of carbon. The normal stable form is carbon-12. See, this one has 8 neutrons while the carbon-12 has only 6. So it's unstable and it decays at a certain rate. When it decays, it emits particles and it loses actually that neutron. Let's say that neutron transforms into a proton. Now, if an element changes number of protons in its nucleus, it's still the same element, becomes a different element. So that's what happens with carbon. It becomes nitrogen because it has acquired, it's transformed, okay? This is complex stuff, but you see that from 8 neutrons becomes 7, and from 6 protons becomes 7 protons. So it becomes a different element. This is nitrogen.
Would it gain another electron? A new element?
Yes, because it's a matter of like how these particles can literally split, can break apart and become different particles. Okay, so we go into advanced chemistry. I'm far from giving you a chemistry lesson today. Okay, but so long story short, okay, there is this decay. There's a lot we have to go through. This is not the important part. Okay, I'm not going to ask you this in the test. I can't explain to you how we do the radiometric dating without at least, informing you of all this. So there is this decay from carbon-14 to nitrogen-14 in the decay of carbon-14. And we take advantage of this knowledge to do our radiometric dating. Now, couple of more things you must understand for now. Every one of us in its body has all organic molecules. All organic molecules contain carbon. But the most abundant type of carbon in our molecules is carbon-12, which is the most common form that is found in the atmosphere. All the carbon that ends up in our body, where it comes from, ultimately, think about the process. The plants uptake a carbon, from the atmosphere, so it's all there. Now, the most abundant carbon in the atmosphere is carbon-12. There is a little bit of fraction of carbon in the atmosphere that forms of that crazy isotope Let me show you this one. And actually, but don't worry too much about the details. It's like when the cosmic radiation hit nitrogen 14, nitrogen 14 becomes carbon 14, and then it becomes nitrogen 14 again by decay. Okay, it's like giving and taking out energy. But anyways, long story short, there is a certain fraction of carbon 14, the crazy cousin in the atmosphere, which ends up in all organisms on Earth, plants, and the whole food chain, right? Because when a photosynthesis process happens, it doesn't distinguish between the carbon 12 and carbon 14, still carbon, okay? So every one of us, every living organism, has a small percentage of carbon 14 in the body. Now, these we can refer to a number of atoms. Now, if I am an ant or a human being, it's different, right? The ant will have far less carbon 14 atoms than me, right? So I'm much bigger. So depending on how big the mass of the organism, there are a certain number of carbon 14 atoms in the body. The bigger the size, the bigger the number, as you can imagine. Now, throughout the life of an organism, the carbon 14, the number of carbon 14 atoms stays constant because there is constant replacement of molecules, okay? And again, depends on the weight. As an organism dies, the carbon 14, that number of atoms of carbon 14 starts decaying because there's no more replacement, the organism is dead. And starts decaying at a very constant, specific rate. So this is what you need to understand. And of course, we learned that carbon 14 decays into nitrogen 14. Now, the other important thing to understand is that the rate of decay is constant, and there will be, as you can imagine, a point in time where half of the atoms of the crazy atom, the unstable, have actually stabilized, have actually lost that energy and have become the stable isotope, right? That moment in time, when that happens, is named half-life. Half-life of the unstable isotope, and the half-life doesn't change because it takes always the same numbers of years for that initial number of carbon 14 present either in an ant or in a dinosaur to become half. Doesn't matter the quantity you start with. The time for that quantity to become half, to decay halfway, is always the same. So it's a constant. Okay, it doesn't change. For carbon, the half-life is not very long, actually, believe it or not. It's only about 6,000 years. Sounds like a long time to you, but it's not very long in geological terms. Something long will be something like billions of years. And thankfully, there are some isotopes in nature that take billions of years to decay. Okay? So, therefore, when we want to utilize carbon to date a fossil remain, there is a limit. Carbon has a very big limit. We cannot date anything that is older than about 60,000 years because its half-life is very short. Okay? So there is a limitation. And whenever we cannot use carbon to date fossils, we will utilize other elements, as I will show you. Okay, so I'm going to show you now this video. Actually, I'm going to first finish, okay? And then I'll show you the detail. This is what I just told you. So if you have an organism, you find an organism remains a fossil, could be also, it could be just the remains of an organism, still a lot of organic material there, and you want to know actually when time of death happens. Of course, you can utilize carbon-14. Now imagine visually that this is the amount of carbon-14 atoms present initially in that organism. You estimate this. When you find a fossil remain of dinosaur, you know, it's huge. It's a huge skeletal system, so you can infer more or less how many atoms of carbon-14 were in that body at the moment of death. So you infer that, and then you, with the instrumentation, you can detect how much carbon-14 is left. And you know that you have a value of referment because you know exactly what half-life of each isotope has been calculated, what it is. And so the half-life for this amount of carbon-14 will be the moment at which half of that has transformed into this blue, which is nitrogen. Okay? Now, I already told you all this already. So while the organism is alive, the carbon-14 content is constant. As the organism dies, it starts decaying. I told you that, unfortunately, with carbon-14, because of the short half-life, only about 6,000 years, precisely 5,730 years, we cannot date things that are older than about 60,000 years. All right? Let's watch the video now. And then we'll do a little exercise on this.
Meet Stitch Fix. Get styled in 4 easy steps. First, take your style quiz. You'll get a box.
How do we know how old something is? For people, we'd ask to see their birth certificate. For trees, we count the rings. But how do we know how old a fossil is? Fossils have their own internal clock. Scientists can read it by looking at the ratio of 2 different types of carbon atoms. Of course, every living thing is made of carbon. Plants grab carbon dioxide from the atmosphere and use it to form complex organic molecules. Animals get their carbon by eating these plants. But there's more than one form of carbon. Most carbon atoms have 6 protons and 6 neutrons. We call this carbon-12. High up in the atmosphere, sometimes cosmic rays hit nitrogen atoms. This creates carbon with 6 protons and 8 neutrons. We call this carbon-14. Carbon-12 and carbon-14 behave alike, but carbon-14 has one unique and important attribute. It's unstable. So once an animal dies, the carbon-14 in its body will start to go away. Every 5,730 years on average, about half of the carbon-14 atoms will decay into nitrogen. This is its half-life. After one half-life, the animal will have about half the amount of carbon-14 it started with. After another half-life, it will have about a quarter. And after another half-life, it will have about an eighth. By contrast, the amount of carbon-12 it has in its body will stay the same. By measuring the ratio of carbon-14 to carbon-12, we can measure how many thousands of years have passed since the animal died. Carbon dating works for fossils up to about 60,000 years old. For older fossils, scientists use unstable elements that have much longer half-lives. For Scientific American's Instant Egghead, I'm Michael Moyer.
Okay, so I guess what I told you, in case you didn't understand what I told you, it explains it really well. So, all right, so I wanted to do a little exercise. So imagine that you have discovered a new fossil remain. And based on its mass, we can infer that it had about 1,000 atoms of carbon-14 when it died in its body. We measure the amount of carbon-14 that is now remain there. And imagine that we measure that there are 125 atoms of carbon-14 there. And we need to infer how old is this fossil, when did it die. So we can go in a stepwise process. And so when the animal was alive, or at the moment of death, you can say, we infer that there were 1,000 atoms there, based on the mass again. And we set that time as 0, that's the time of death. Remember that we have detected what the problem told you is 125, right? Atoms. Imagine if we had found 500 atoms. What would that tell you? How old is that remain?
6,000.
About the half-life, which is about 6,000, okay, which is precisely 5,730. 6,000 is good enough, okay? Just try to remember, for me, that is about a few thousand, not billions of years or hundreds of thousands. Just try to kind of have that kind of thing, okay? I'm not asking you too much detail. But it's not 500. Imagine that we had found 250. What would we have to add here to 5,730? Another half-life. So that would be, but we found 125 atoms, so you have to add another half-life. So that's how we would kind of like calculate the number of years since then, okay? All right, so now as I told you, geological times are much more than 60,000. We know that life started on Earth about between 3.5 and 4 billions of years ago. So if we find fossil remains that theoretically date back to 3.5 billions of years ago, we obviously cannot use carbon. There will be undetectable carbon-14 there. In that case, we can refer to other isotopes, and these are isotopes that are not present in organisms, but they will be present in the minerals that have substituted all the organic material, the minerals from the rock that have come into that shape, okay, that we look at, as I told you, when we look at a fossil remain formed by mineralization. And therefore, we can utilize, for instance, uranium-238, look at the half-life, billions of years. So theoretically, if we find one of the first organisms that appeared on Earth, maybe bacteria, do you think bacteria can fossilize? You're correct, okay? And we're going to talk about fossils of bacteria in a minute here. And so they will be billions of years old, and therefore they have been dated by using, for instance, uranium-238. So as uranium, there are others, thorium, rubidium, others that experts can use. And again, all the half-life values are fixed values for each of these radioactive parent elements. And here it shows you the daughter product, the stable daughter product that forms after decay. I don't mean you to remember any of these. Just try to remember for me that there are elements other than carbon, of course, that we use for dating fossils. Otherwise, we'd be out of luck because we couldn't date anything that is older than 60,000 years. Okay. All right. So this is just a summary again of the comparison between relative dating and radiometric or absolute dating. One uses the order, the strata in the rocks to determine the relative age of fossils. The other is a fairly precise methodology based on half-life of radioactive isotopes. Okay, so the summary of all of this, okay, is here. I have a question for you. How old are you?
22.
You sure? That's shocking news. You are about 3.7 billion years old. You know that? So, we have found some interesting rock formations on Earth known as stromatolites, spelled here. The rock formations that have these bluish lines, you cannot really see them very well here, they're bluish lines embedded in them, which are fossilized bacteria. And they are fossilized photosynthetic bacteria. Bacteria that are green bluish, okay, and do photosynthesis. We found this rock formation, we date them using uranium, and they date back to about 3.5 billion years ago. So we know these organisms were present on Earth at that time. And these special bacteria, greenish bluish, they're called the cyanobacteria. You have to remember this name for me from now and until the end, until the last hour. Then you take the last test, then you'll forget it, right? Anyways, it's very important. Okay, I'll tell you in a minute why these organisms are the reason why you are the way you are today. They're the reason why you have evolved the way you have evolved. Okay? Evolution could have taken a very different route if these organisms had not appeared on Earth. Okay? So, definitely represent a tipping point for evolution on Earth. So now, these cyanobacteria, are they extinct? No, they're still present on Earth very abundantly. And these rock formations, you can still find. They start like muddy things in which, it's mud and bacteria growing in between the mud. And then throughout the fossilization process, they become hard rock. Okay? So they're still forming nowadays, and they will become fossil in so many millions, billions of years. So the name of the rock formation is stromatolite, and the specific group of bacteria, this photosynthetic bacteria, is cyanobacteria. You must remember for me, stromatolites, the oldest stromatolites date to 3.5 billion years ago. This is just a schematic representation, okay, where it shows you how all these green bacteria grow between the layers of mud and then becomes rock. So these are bacteria. So remember the structure of bacterial cell, prokaryote cell, no nucleus, and these are photosynthetic bacteria. Again. Now, are these the first bacteria to appear on Earth? We don't think so. There is a lot of evidence that although we don't have fossil evidence, in reality, these are not the first bacteria to appear on Earth. These are the first photosynthetic bacteria to appear on Earth. Before these bacteria, there were heterotroph bacteria already present on Earth, lots of species, okay? And one of the reasons why we know this is that when we analyze the ancient atmosphere, we know that there was no oxygen whatsoever in the atmosphere before cyanobacteria appeared, before 3.5 billion years ago. The atmosphere on Earth did not have oxygen whatsoever. The first organisms to input oxygen in the atmosphere were cyanobacteria, because through the photosynthetic process, what is one of the byproducts of photosynthesis? Oxygen, right? We all know that plants are good for us because they produce oxygen through the photosynthesis process. This is the same, right? Same photosynthesis process done by bacteria produces oxygen. And what we think that happened actually is this. As cyanobacteria evolved and started producing oxygen, that gas initially was very toxic for the pre-existing bacteria. It's like now, all of a sudden, imagine that CO2 in the atmosphere becomes like 30%. We all die, okay? So this new gas that was introduced in the atmosphere was extremely toxic. And most of the heterotroph bacteria that were there died because of this gas. But the ones that managed to survive, because they had specific mutations, evidently, they managed to start utilizing it to their benefit. What do we need every single second, every single minute of our life?
Oxygen.
If we deprive our body of oxygen for a few minutes, we're dead, right? Fundamental. And so all other animals. So, what I was trying to tell you before is that the input of oxygen in the atmosphere has shaped the course of evolution, life. Because although it was very toxic initially, the organisms that the other bacteria that managed to survive it started utilizing it eventually in metabolism, and we are the result of that, because we utilize oxygen every single second for our metabolism. That's why it's so fundamental. Without oxygen, our body won't make energy anymore, and it will very quickly die. The cells will die. So, thanks to the advent of cyanobacteria, life on Earth is what it is. Could have been completely different if these organisms hadn't evolved. I don't exclude complex life would have come in a different way. A different type of metabolism could have evolved. Okay, it's kind of fascinating to think about all this, right? All right, so now the other thing I want to tell you is this. You learn in school that plants do photosynthesis, right? That is not true. Plants, a plant cell does not do photosynthesis. A plant cell has stolen a cyanobacterium, and it has eaten out a cyanobacterium and kept it as a little machine that now is inside the plant cell, and of course the plant cell now does photosynthesis. But those organelles, who knows from school, those organelles in plants that in which photosynthesis happens, how are they called? Anybody?
Chloroplast.
Chloroplasts. Guess what a chloroplast is?
And the sun. Come to light.
Is a cyanobacterium, okay? At some point during evolution of life on Earth, a cell has made friend with a cyanobacterium and has become a plant cell. Now it does photosynthesis. But in reality, if you go back and you think about it, only cyanobacteria do photosynthesis on Earth, because a plant cell is nothing else than a cell, a heterotroph cell that has made friend with the cyanobacterium and kept it. Okay? So we'll talk about that very much in detail when time comes, starting next unit, how plant cells have originated, how chloroplasts have originated. If you start remembering it now, it's good. But yeah, plant cells per se don't do photosynthesis. So the cyanobacteria in plant cells do it. Okay, so pretty much already told you all this is philosophical thoughts. Okay? And it should be a short video. This video about cyanobacteria. And then I would like to do a little review here.
Cyanobacteria are photosynthetic eubacteria. 2 billion years ago, cyanobacteria and other photosynthetic bacteria dominated Earth. Today, the descendants of these organisms are still common in ponds, lakes, and oceans. Cyanobacteria are prokaryotic organisms. The prokaryotic cells of bacteria are very different from eukaryotic cells that make up plants and animals.
If we happen to do a little bit of microscopy to look at produce, most likely we will find some of these. And I just wanted to point out, this looks like all these circles are the bacterial cells. What these cyanobacteria like to do is they like to grow in chains, but they're all single bacteria. They just associate in chains. So that's what you're seeing here, all these filaments. It's not a multicellular organism, okay? It's still one cell organism.
Prokaryotic cells do not have a nucleus. DNA molecules of prokaryotic cells are circular in structure. Prokaryotic cells are usually smaller than eukaryotic cells, less than 10 microns in length. Prokaryotic cells are very simple in structure. They do not have organelles found in eukaryotic cells. Prokaryotic cells do not have a cytoskeleton. Cyanobacteria are photosynthetic, capturing energy from the sun and evolving oxygen just like the land plants. In fact, cyanobacteria are responsible for the generation of most of the oxygen in the atmosphere of the young Earth. Cyanobacteria live in a wide range of environments, from extremely cold lakes to hot springs. The hot springs of Yellowstone National Park are a home to various species of cyanobacteria. They are responsible for the orange and yellow color seen around the perimeter of the hot springs. Cyanobacteria often live on the surface of layered mats. These mats were much more common in the geological past, and many became fossilized. Fossilized bacterial mats are called stromatolites. Cyanobacteria have other interesting features. For example, some have modified cells called heterocysts. These heterocysts have the ability to fix nitrogen from the atmosphere to form ammonia. This fixed nitrogen is ultimately available for the growth of the cyanobacteria or for the growth of nearby plants. Another interesting specialized cell of cyanobacteria is the aconite. An aconite is an enlarged, thick-walled cell which is heat and drought resistant. The aconite cells allow cyanobacteria to survive extended periods under harsh conditions. This video has illustrated only a few of the most important.
I'm going to stop here. Don't worry too much about all the details of all the heterocysts that aconite, but one thing that we're going to learn is that these cyanobacteria, of course, as you can imagine, they are made through all events of mass extinction. So they have some forms. You can imagine like that they can freeze themselves in forms that are very, very resistant to harsh conditions, ice ages, and things like that. They were able to survive and eventually come back to life in the cyanobacterium form again. Okay, so I think this one is done. So, is there anything in particular you'd like me to explain again? I can do that first, or we can just go a very quick review of all the lectures. Okay, so first lecture, science of the process, okay? So it's pretty straightforward, right? We all kind of like have heard about this already in school. Flux is a very straightforward logical process. These are the steps necessary to go through if you want to prove an hypothesis in science. So you have to set, first you set your hypothesis, then you have to set an experimental procedure that makes sense, where you can get data numbers, then you analyze your data and make your conclusion, right? So remember, in validating hypothesis, you cannot go in any other way than just set up an experimental procedure using numbers. Only numbers will tell you if your hypothesis is true or not. Remember, we gave many examples of studies that we can do, hypotheses that we can test. We talked about testing drug, and then we talked about something that seemed really difficult, a question that was, to me, seems very difficult to answer. Even in those cases, you can still do experiments and come up with numbers. The one thing that we need to able to define and recognize is what's an hypothesis and the difference between hypothesis and theory. Remember, hypothesis is very narrow in scope and is a tentative statement to explain a phenomenon as limited evidence needs to be proven. While a theory is something completely different, as we'll see in a minute. Now, we talked about several experiments, testing several hypotheses. The one important thing is to define in an experimental procedure is to define the independent variable and the dependent variable. Now, I may, for instance, give you examples that we haven't talked about. I may give you a scenario of an experiment and ask you, what is the independent variable in this experiment and what is the dependent variable, with choices. So if you have understood the concept, you should be able to recognize them in new examples, right? It's not that we have to talk about always the same examples that we get in class. So, concepts here, okay? Independent variable is the factor that you're testing and it does not change. And the dependent variable is the factor that you're measuring, so that can change, right? And in every experiment, the idea is that the independent variable is causing a measurable change on the dependent variable. So these are the main points. And I wanted to mention, go over again, that question, this question, okay? Or this hypothesis introduced by Henry Bates about evolutionary mimicry that he hypothesized that evolved in order for, see, I'm being teleological here, evolved as a result and to avoid predation, okay? So what did he do? This is a biological question. Remember, we must come up with an experimental procedure and numbers. Remember what he did? Pick snakes, okay, put them in the field, and then counted the number of bites, which is the number of predation events, if you will, okay? And then he collected them back, and he put together his data. And of course, in his experiment, he had a control. Without a control here, if he didn't have a control, and remember he was testing two different areas, without a control, even if he had this huge difference of predation events on the striped snakes, the ones that resemble the bad guy, remember, he would have not been able to understand if the difference was due to a different number of predators. Okay, so control is always fundamental. Okay, and you must always have a control. Now, the other important thing we talked about here, okay, is how do we recognize if the data, see the difference that we see, like in that graph, is enough, is big enough. Sometimes you'll see very small difference and you won't believe that there is a real difference there in your results. So there must be, again, something objective, like a number that tells you if that difference is true or not. And so that number, we call it p-value. It's a number that is obtained through sophisticated statistical analysis. Usually you use a program, you plug in your results in the program, you tell the program what kind of statistical analysis you want to do. Maybe it's a comparison between only two groups, maybe it's more than two groups, so there are different analyses you can do. And the p-value represents the probability that your results have been obtained by chance, like your chance. So you want that number to be very little. You want your statistical analysis to be significant. And so the cutoff number that has been established by mathematicians and statisticians is 0.05. Okay? Basically, there has to be less than 1 in 20 chances that your results have been obtained by chance. If that is the case, then your results are considered true, significant. Okay? So that's what we do. All right? Again, sometimes you have very small difference between two groups, and then you do statistical analysis and will be surprised that it could be significant. It all depends also on the size of your samples. If you have collected a lot of samples, you have a lot of data, even with a small difference in your results may be significant. So you just rely on the p-value. That's it. No opinion here. Okay, then we define theory. Okay. Theory is something much broader than an hypothesis. A statement that describes a natural phenomenon has been recognized by the scientific community for a long time. Sometimes theories can evolve, can become more comprehensive, okay? But typically theories are true, or at least on Earth, maybe on another planet. Some physical theories will be different, but at least the theories that we recognize nowadays, we can be sure they're really true. Okay, so that was it for this lecture. I know I'm going fast, but I'm assuming you guys have already kind of studied, right? And if you have questions along the way, please let me know. All right, so in this second chapter, we went over a checklist of characteristics associated with life, although there is only one thing that defines life, which is the one thing that defines living organisms.
Carbon.
So CO2 in the atmosphere is a living organism. Has carbon in it. Guys, no
Cells.
Oh, thank you. Cells, okay? I told you many times, so we went through over this chapter. So the only thing that defines a living organism is the cell. If you're not made of cells, I don't care that you can reproduce, I don't care you have DNA or whatever else, you're not a living organism. Do we know of something like that? Viruses are not considered living, believe it or not, okay? All right, so the cell defines living organisms. Now, these are all characteristics associated with life, okay? Not that they don't define life per se. All right, so we went through the levels of organization. I'm going through the summary slides right now. You have to know this order of hierarchy here. From biosphere, ecosystem, and the definition, okay? Biosphere are all the ecosystems on Earth. Ecosystem are all the organisms plus the non-living things. Remember the factors that sustain the organism, the abiotic factors. Community, take away the abiotic factors from ecosystem, you have a community. So all species living in a certain place. Population are groups of the same species. Then you arrive to organisms. This is the upper tier. and you have a window here. I'm going to the summary slide again. So from organism, in an organism you can recognize an order of complexity, especially talking about multicellular organisms, obviously. Some organisms won't have this, would have just the cell, right? The unicellular organism, that's the highest level of complexity. You go from organisms to organ systems, organs, tissues, and cells. This is the basic unit at which we recognize life. After that, there's no life. Or the pain is not considered living. Okay, we can zoom in even more inside the cell. Okay, to subcellular compartments, organelles, particles, and then of course molecules and atoms. Okay. Then we went through all the other characteristics. Before I move on, I just want to make sure you know this slide very well. Okay, all living things are made up of one another. So this is what defines really the living. Okay. Please don't forget. We talked about other characteristics of life, such as regulation. We talked about other some examples of feedback mechanisms to maintain the constant conditions in organisms, even if they're complex or they're simple. At the end of the day, a complex organism, multicellular organism, wants to maintain the constant environment of the cells of which it is made. So there are the systemic feedback mechanisms that are aimed to maintain the cellular homeostasis, we call it, okay? Internal and external. We talk about temperature, we talk about water content, whatever you want. And the other characteristics, such as response to environment, energy processing. And regarding energy processing, you have to know, of course, the definitions of the different types of organisms that carry out different types of metabolism, and in regard to if they are able to make their own food in terms of glucose or not. So autotroph and heterotroph, respectively, right? These are very important terms. Some of you may remember them from school, I'm sure. You have heard, if you have forgotten them, you have to learn them again. And then we recognize different categories, you see, of organisms in food webs. Producers are the autotrophs. Are they only plants? What did I tell you? Actually, plants haven't invented anything. The only invention was by cyanobacteria. So we know now there are also bacteria. They play a very important role as producers, but also many produce, as we will see. Consumers and decomposers. We are consumers. A special category of consumers is decomposers. Decomposers attack and break down, we can say, the complex organic matter into simple molecules, carbon molecules, nitrogen molecules, all elements that are fundamental for organic molecules to be taken again by the producers. And so there is a recycle. The reproduction, growth and development, it's pretty straightforward, and evolution. Okay, so we talked about taxonomic classification. The three-domain classification system is a must know. And what is the characteristic upon which we distinguish the three domains? My previous class couldn't answer this question, and I'm very sad because I said so many times. Come on, guys, you know it. You're overthinking. What is that thing that makes me distinguish the three domains?
Is it the DNA in or outside the nucleus?
Say it better. I mean, I kind of understand you're on the right path, but it's not an acceptable question. An acceptable answer. Who said something? I couldn't.
I said the sequence.
Dependence of the nucleus?
Which means
Oh, eukaryotic or prokaryotic?
cell type. There are two types of cells, right, in nature? The prokaryote cell, the eukaryote cell, right? So that this distinction is made upon the type of cell. Simple as that. Why I remember saying it so many times, okay? So when I say something so many times, it's a clue. You must know it. Right away, study it so you don't forget and you know it for the test. So, okay, so going to the cell type to explore. So it's true that one has nucleus, the other doesn't, but also there are also other differences, okay? The main thing is the presence or absence of nucleus, but for instance, prokaryotic cells, remember, they're smaller, okay? At least 10 times smaller, and also they don't have any subcompartmentalization. Remember, a cell is like a factory. If the factory is a small factory, maybe things can kind of be, maybe there's not much space to create facilities around, okay? So maybe things are more floating around in the same space. But if you are a huge factory, a huge production there, you must kind of like be very organized. So there will be a building for production of energy, a management building, a dispatch meeting because you are producing things and maybe are exported outside. So think about cell in this regard. We'll obviously go over this, all the cell components, okay? But this is very important to know, okay? Then we went and we touched on like the various characteristics of the domains, domain bacteria. What is the, why do we distinguish archaea from bacteria if they in fact are made of the same type of cell? Both are prokaryotic, but why do we distinguish them and we don't put them in the same group? Is there anything special about archaea or different?
Can archaea survive in more extreme environments?
They're extremophiles, right? We say. So they love extreme environments, various. Okay. Then we said, okay, now eukarya is a huge domain. It comprises many different types of organisms, plants, animals. So we need to subclassification. 4 kingdoms, must know. Must know. Okay. And then we touched on the various kingdoms. The kingdom Protists, I really would like to do a little microscope activity. Starting next unit is kind of like the first group that we're going to look at. I do have the sample at home. They're still alive. I'm keeping an eye on them every night. And hopefully, we can remember them a little bit better. So we talked about protists mostly unicellular. This unicell is one cell, okay? I cannot tell you where the nucleus is. I cannot tell you, when I look at the microscope, which one is nucleus. There are so many vacuoles, so many compartments. It's impossible to tell which one is the nucleus unless you fix it and you stain it. You stain it with a substance that will highlight the nucleus. Then you can tell, okay? When it's alive like this, you can't really tell. It could be this dark. All right. So, anyways, then we talk about fungi. What's the characteristic of fungi? Are they autotroph or heterotroph? Heterotroph, absolutely. But they look a little bit like plants. They don't move. So there's a kind of between characteristics. We put them in a kingdom by themselves. What's the peculiarity of fungi? Anybody remembers the special thing they do? It's written there anyways. They do a type of digestion known as extracellular digestion. They kind of produce enzymes, they release them outside. Those enzymes will break down the dead matter. They will absorb the broken down molecules. Plants, okay, and then plants, of course, they're photosynthetic, but we learned today that they have not really invented photosynthesis. Right now, you know something more than before, or something more than the average person out there, right? Okay, you know, thanks to what plants do photosynthesis. Now we talk about animals. Animals, they're all consumers and they move at least at one stage of their life cycle. We learn how taxonomy works and how we name species. Remember the binomial nomenclature. So for instance, again, my test is not so much about like how do we do the nomenclature of species. I just give you a name and I ask you, is this a species? So if you know how species are indicated, you should be able to answer, right? A little bit step ahead of just definition. Why these three very similar organisms are different species?
What was the question?
Why these three very similar organisms are three different species, right? There are three different species, do you agree? Okay.
They can't produce fertile offspring.
Okay. Either because I don't know if a dog would ever mate with a coyote. I don't know, it's possible. Maybe a dog that is grown more like, grew up more in like
There's wolf dogs.
wild,
Wolf dogs.
yeah. They possibly mate, but I don't think they produce fertile offspring or they produce offspring. That's why they are considered three different species. So there is a block at the beginning where they don't mate. They may mate, you know, sometimes weird things happen. I don't know, here is the next lecture. Remember when we talked about these hybrids that are sterile, they are viable, they give a long life, but they're sterile, okay? It's like almost there. Okay, the two species are so close, they can produce viable offspring, but the offspring is not fertile. Okay, so definition of species I'm going to skip, therefore we pretty much defined it. Now, hierarchy of taxonomic classification, unfortunately you have to memorize it for me, but remember those acronyms that I gave you, they should be helpful, right? And we gave some examples here of how classification works, okay? And then we talked about phylogeny, okay? Phylogeny is the representation of the history of evolution of a group, okay? And phylogeny is based on characters. Characteristics can be anything you can think about, anything under genetic control, it can be even behavior. We know phylogenetic trees have been historically built upon physical characteristics, anatomical characteristics, or we can say morphological as well. But nowadays we rely a lot on molecular analysis, right? DNA analysis. And remember how we name certain things in phylogenetic trees, certain items. These are the branches, okay? And that infer the time of appearance of the group on this kind of phylogenetic tree. These are the nodes, which are the intersections. Basically the time of the split, okay, between two groups. And this is the root. We talked about clade. Remember, this is a whole clade. Also these, okay, and the tip of the branches are clades as well. This is the whole vertebrate clade, right? This is a one clade, all vertebrates, okay? This phylogenetic tree tells you a little bit of detail how we make the how we consider these different groups based upon which element. Here you have vertebrae, okay, and then you have a split between the formation of bone or non-formation of bone. So that group has vertebrae, has something supportive, but it's not really bone, okay, and so on. So I think everybody can visualize this phylogenetic trees pretty easily, right? Just diagrams. We looked at the tree of life, where all life is represented, and how we understand close relationships. Just go down between groups, and we see there is common ancestry. The line connects basically. We talked about a phenomenon that happens quite a bit in nature, the evolution of analogous structures. Give me an example of analogous structures, if I ask you. Anyone? Analogous. We talked about convergent evolution. Anybody? Come on quickly.
Like wings in a bat and wings in a bird? Or no?
Those are homologous because there are bones, a set of bones. What about the wings of a butterfly and the wings of a bird? Can you recognize bones in the wings of a butterfly?
No.
It's so distant, so far away from a bird or a bat. Okay, so those structures have evolved independently just because the environment has put a pressure. Or we learned about how whales evolved, right? Where did a whale evolve from?
The sonics or the wolf.
A wolf-like creature, a tetrapod, okay? Not from a shark or from a fish. So it's like going back to a shape or acquiring a shape that is ideal for an environment, like the aquatic environment, which is very similar to the shape of a fish, because that's the ideal shape for that environment, okay? So it's not that the two groups are very closely related. It has been a phenomenon of convergent evolution. Evolution has put a pressure to converge to the same shape. But there is no direct common ancestry. If there is direct common ancestry, how do we say those structures to be? Not analogous, but homologous. Those terms you have to know for me, okay? Okay, I guess, okay, so of course the chapter of evolution is very extremely important, okay? It's more conceptual, but the observations and the deductions that Darwin made are extremely important to know. Okay.